Hello to our friends joining us via recording. We are working through the second part of lesson number 13 about the special senses. To get us ready to talk about the rest of special senses, we did a review of the process of vision. So to help us out with that, to begin with, we looked at a picture of an eyeball to remind us of some of the most important parts of the eyeball in the process of vision. So we're going to work our way from front to back, the way that light waves would travel. Let's start with this first part right here, this clear part on the front of your eye. What is the name of this clear part on the front of your eye? Yeah, so that front part of your eye, the clear structure in the front is called the cornea. When we talk about the cornea, this is one of the structures in your eye that does something in particular. What does the cornea do? What's its main function? Oh, I know we know this. There we go. The cornea is the first place in the eyeball that's going to bend our light. Specifically, the cornea bends the light to take it from whatever direction it's coming from outside the eyeball to get it in so that it can go on to my second structure that bends the light. And that second structure, like I'm seeing us mention here in the notes or in the chat box here, is the lens. So the cornea is on the very outside. It takes light waves coming in at whatever angle and it bends them so that they hit the lens. The lens is also going to bend those light waves. Where is the lens trying really hard to bend those light waves to? Where's the lens trying to focus the light waves? Yeah, so specifically, the part of your eye where you can see the most clearly, which is going to be our next label here, is a place in the back of the eye called the macula lutea. And we mentioned during our group work that that is a lab structure. So we did mention it from lab. It may not have been quite as prominent in lecture, but the macula lutea, this is the place on your retina where you see the clearest, so where we can see best. That's because the macula lutea is the home to our type of visual cells that see really well in bright light or that see in color. Which of those types of visual cells do I have a ton of right here in the macula lutea? Yeah, it's those ones that are called cones. So the macula lutea, it's where we see best because we have the most cones. This is the place in the eye where they are the most concentrated. So the goal of the lens is to get light waves onto the macula lutea where I can see them most clearly. That's because that's where I have the most cones, my cells that see really clearly. In order for the lens to be able to focus it on the macula lutea, that light that came in had to be bent by the cornea to get onto the lens. And the other thing to help us make sure we focus in the right place is we have to use these structures that I see on either side of the lens. What's the name of these things right here? Yeah, so those things with the last labeling thing is called the ciliary muscles, or we sometimes call them the ciliary body. The job of the ciliary muscles or the ciliary body is to change the shape of the lens. So they can pull on the lens or they can relax the lens, depending on whether you're seeing something up close or something far away. We have to change the shape of the lens to be able to get those light waves back to the macula lutea. So the job of the ciliary muscles is to change the lens shape. Again, to help us focus our light onto the retina, the particular part of the retina called the macula lutea. Remember our big picture idea from class last time. Light waves must be focused on the retina for us to be able to see them. If I focus my light waves in front of the retina, if I focus them behind the retina, if I focus them in two different places in the eyeball, I won't be able to see. So light waves must all be focused on the same place in the retina, preferably this place called the macula lutea. Focus them there, which allows us to see very clearly. We talked about several vision disorders last class. All of them have to do with us not being able to focus our light correctly at a particular place in the eyeball, on the retina. There are lots of different reasons that we might not be able to focus that light. So we're going to outline the reasons that we can't focus the light on the eyeball. And we're going to talk about which kinds of light don't get focused or what's going on with that light. And then we're going to remind ourselves if this is something that you're born with or something that you develop over time. So I'm going to kind of flip this on its head. When we were working in our group, we worked our way straight down the table. I'm actually going to be mean and make us work our way across the table, mix it up a little bit for us. So we're going to start with a quick description, basically in easy person words of what's going on. So if you have someone who comes up to you and they say, I have hyperopia, what does that mean about their vision? What can they see? What can't they see? What's going on with hyperopia? Yeah, so hyperopia is the anatomy word, the fancy word for farsightedness. Farsightedness. If I say that someone's farsighted, they can see far away. That's what farsighted means. Means they cannot see close. Farsightedness. Okay, so someone who comes up to you and says they have hyperopia, they are farsighted. Who else, which other vision disorder would we say makes someone farsighted? There's another one on this list that we can see far away, but not up close. Yeah, exactly. The other one that we would say is farsightedness is presbyopia. Presbyopia. Now, there's a difference between hyperopia and presbyopia. What was the phrase in class last time when we were talking about presbyopia? We said something in front of farsightedness. Does anyone remember what was the phrase that we put? If you have presbyopia, you have, yeah, age-related farsightedness. Regardless of whether you just have farsightedness or whether you have age-related farsightedness, the problem is still the same. You can see things that are far away, but you can't see things that are up close. Now, what if I am nearsighted? What's the name of the condition if I'm nearsighted? I can see things up close, but not far away. Yeah, that one is called myopia. Nearsightedness is myopia, meaning I can see close, can't see far away. Nearsightedness. I can see what's near to me, but not what's far away from me. There's one other vision disorder here on our list, and it's not nearsightedness, it's not farsightedness. When we think about astigmatism, kind of think about astigmatism as everything's blurry, basically. It's not just if the light wave comes from far away. It's not just if the light wave comes from close. In astigmatism, everything's blurry, regardless of distance. What is it in astigmatism? What was going on with the light that was problematic in astigmatism? We had it listed before on our table. Yeah, so the problem with astigmatism is we have, in our technical anatomy speak, multiple focal points. Multiple focal points. That's a fancy word for saying multiple places that light goes. Multiple places that light goes. We need our light to be focused in one place for us to be able to see it. We showed on our previous picture, the one place we need it to be focused is the macula lutea. If I don't focus my light on the macula lutea, I can't see it. Sometimes I can't see the light that's close to me because I can only see things far away. Sometimes I can't see the light that's far away. I can only see what's close. With astigmatism, I can't see any light because all of my light is coming in and focusing on multiple places, and none of those places are the retina. So astigmatism is a little bit different from the others in that it doesn't matter if it's up close or if it's far away, I can't see it. It's blurry because I have multiple focal points. Now, when we talk about astigmatism, what is it that makes us have multiple focal points? What's my anatomy, or what structure in the eyeball is wrong to give me astigmatism? Yeah, astigmatism is a problem with the cornea. So the cornea has the wrong shape. If the cornea has the wrong shape, remember how we talked about that's the first place that bends light. I bend the light onto the lens to bend it toward the retina. Well, if I mess it up at the beginning and it comes in the cornea wrong, that's going to give me issues with bending it with the lens, and I end up with multiple focal points in the eye. So astigmatism is a problem with the shape of the cornea. When I talk about hyperopia and myopia, in these disorders, it's not an issue with the shape of the cornea. In these disorders, it's an issue with the length of the eyeball, right? So these are a problem with the length of the eyeball. When I talk about someone having hyperopia, what's going on with the length of their eyeball? Hyperopia. Yeah, in hyperopia, the eyeball is too short. The eyeball is not long enough. So when I go to focus light waves, especially those light waves that come in from really close to me, my eyeball is too short for me to be able to focus them. They come in too fast. The eyeball, or when my lens tries to focus them, it ends up behind the retina. I send them too far back. If my eyeball is too short, light waves travel too far. They go behind the retina. Behind the retina. Hyperopia, an issue with the eyeball being too short. What's the issue with myopia? What's going on with myopia? Yeah, the eyeball is too long now. It's the opposite. So my light waves don't travel far enough, which means that they end up in front of the retina. And remember, we were talking about last time, if my light waves are in front of the retina, I don't have rods and cones in front of the retina. If my light waves are going behind the retina, I don't have rods and cones behind the retina. So I'm not going to be able to see light waves that focus behind or in front of the retina. That's not going to help me out. Now, when I talk about presbyopia, age-related farsightedness, this is not actually an issue of how long or short the eyeball is. In presbyopia, I'm having an issue with a couple of the structures in my eyeball. What are the two structures that give me trouble when I have presbyopia? Yeah, so the first structure that's going to give me trouble in presbyopia is my lens. When I talk about the lens, which I use to focus the light on the retina, as we get older, the lens gets harder, or I'm going to say it gets stiff. Because remember, when you go from seeing up close to far away, you have to change the shape of the lens. When my lens gets stiff, it's harder work for me to go from looking far away to up close, to change the shape of the lens. The thing that's supposed to help me change the length of the lens are the ciliary muscles. Their job is supposed to change the length of the lens. Well, the problem with the ciliary muscles is the older we get, the weaker they get. The ciliary muscles get weaker. If the ciliary muscles get weaker and their job was to pull on the lens, and the lens gets stiffer or gets harder, I'm really not going to be able to focus my light that comes in this way. So age-related farsightedness has to do with that lens getting harder and the muscles that change its shape getting weaker. That makes it so that I can't see light. Light still ends up behind the retina, but it's not because the retina is in the wrong place. It's because I can't move things the way that I normally would. I can't bend my light the way I normally would. Our last thing to know about these disorders is when they develop. Really, the biggest thing you need to know about these disorders is there's just one of them that only is seen in older people. Which of my disorders is the one that you only see in people who are over 35 years old? Yeah, so the only one seen, so only seen in people who are 35 plus. Ooh. Number 5, not quite. 35 plus years old. The only disorder that you can't be born with, basically, is presbyopia. Presbyopia is something that you grow into. And like I mentioned before, pretty much everyone is going to grow into it. So between the ages of 35 to 50, naturally what happens in our bodies, our lens gets stiffer, our muscles get weaker. Everybody is going to develop presbyopia. Hyperopia, myopia, astigmatism. You could be born with any of these conditions. You could be diagnosed with any of these conditions in your childhood. I think I mentioned this last time. I was diagnosed with myopia in 1st grade. So myopia can develop very young. Very young children, if you see them in glasses, probably they have hyperopia. So these are the kinds of conditions, or astigmatism as well. You can be diagnosed with that as a child too. So these other eye conditions can develop when you're younger, but presbyopia is the exception. We only see that in people who are 35 years old or older. Gloria asked the question in the chat, what we would say about the light when we talk about presbyopia. Where does the light in presbyopia go? Are we focusing light in front of the retina or behind the retina in presbyopia? How would I fill in that part? Yeah, so I'm still sending my light behind the retina. I'm still sending it behind the retina. I'm not sending it behind the retina because the eyeball's the wrong shape, but I am sending it behind because the stuff that was supposed to help me get it in the right place on the retina is too weak to do it. So yeah, this is another issue with the retina, with me sending my light waves behind the retina. When we talk about astigmatism, I send it to multiple places. When we talk about myopia, it's sitting there in front. So with my other disorders, I'll just add a quick note. When does this disorder develop? We'll just say children here to make it clear for ourselves. We could see this in children. Same thing with myopia, we could see in children. Same thing with astigmatism. We could see that in children. We can only see presbyopia in adults. How do we feel about vision disorders? Thumbs up, thumbs down. Send me an emoji in general. Jacqueline's feeling good about them. Awesome. Send you our daily penguin. Perfect. All right, well, let's go to the process of hearing then. We'll dive into hearing. When we talk about the process of hearing, just like with vision, the way that you hear things is because of waves. So the way that light, or that, excuse me, now we're talking about sound. The way that sound travels in the air around you is in the form of a wave. Depending on how quickly the wave travels, whether it's more slow or whether it's more fast, depending on how quickly that wave travels, that helps you to determine the pitch of that sound. So when my daughter's screaming, because let's be real, that's what 4-year-olds do, we've got a really fast going sound wave here. When I'm listening to the hum of my refrigerator, it's going to be a more low-pitched sound, so it travels more slowly. I mentioned this with light, and I'll say it again here with hearing. I'm not going to ask you about high pitch versus low pitch, so about the way that they travel, but I want you to understand as we start talking about the way that we detect sound waves, that different places inside your cochlea detect sound waves that are traveling at different rates. So one place in my cochlea would listen for these really slow sound waves, and a different place in my cochlea listens for these very fast sound waves. So the pitch of a sound, whether it's high pitched or whether it's low pitched, that's going to dictate where in the cochlea we hear sounds. So hearing, the process of perceiving sound waves. We know from lab that there are 3 parts of the ear. So we have the external parts of the ear. These are the ones on the outside of your body. So including things like the auricle, which is the big funnel part of your ear, that funnels things into the ear canal, or in lab, we call this the external acoustic meatus, the tube. Sound waves come in this tube and they bounce off of this place called the tympanic membrane, where we have those 3 little bones called the ossicles. The ossicles take those sound waves and bring them into the internal ear. The part of the internal ear where we're going to do hearing is the part here called the cochlea. So the snail shell part is where we do hearing. Sound waves go into this snail shell part, we hear them in here, and that allows us to process them. Here's a picture that we talked about in lab. You saw it in the lab guided lesson. Here in lecture, it becomes even more important. So this would be an underline highlight star picture because it's important for us to understand how hearing works. Specifically, what I want us to focus on right now is what's going on in the cochlea when we're hearing things. So remember, sound waves come in, that external acoustic meatus. They bounce against this drum called the tympanic membrane. The tympanic membrane is attached to the malleus, which is next to the incus, next to the stapes. All of these parts start rattling with those sound waves. And these ossicles, these little bones, their big function in the process of hearing is to actually make those sound waves louder or make them stronger. Because up until this point, when we hit this structure called the oval window, where we go from the middle ear to the internal ear, up until this point, sound waves are always passing through the air. Once we get into the internal ear, now those sound waves have to start passing through liquid. There's a special name for the liquid that fills the internal ear. There's 2 kinds of liquids, actually. The first one that sound waves go into is something called perilymph fluid. Perilymph fluid. So right on the opposite side of the oval window is a fluid-filled tube called scala vestibuli, or some of your videos will call it scala vestibuli. You don't have to be able to say it, you just have to know what it looks like. So I'm going to call it scala. Scala vestibuli is a tube that's filled with something called perilymph fluid. When my sound waves get into scala vestibuli, they're having to move now in perilymph fluid. It's kind of like when you get into the swimming pool and you start trying to walk forward, how it's a lot harder to walk in the water of the swimming pool than it is to walk on the land on the outside of the swimming pool. So the job of the ossicles is to make those sound waves really strong, or to amplify them, so that once they go into this perilymph fluid and they have to start moving back and forth in fluid, they're stronger. So we cross the oval window, get into the perilymph fluid here in a region called scala vestibuli. At this point, my sound waves are trying to be heard. So they're going to travel back and forth in this fluid up here in scala vestibuli until they find the place in the ear where they're supposed to be heard. So if we have a sound that's really high pitched, so like I mentioned, my daughter's screaming, that's going to be heard up closer to the beginning of the cochlear duct, compared to something like the fridge humming. That's going to be heard a little bit farther in the cochlear duct. So my sound waves travel and travel and travel until they find the place in the cochlea where they would be heard. At that point, those sound waves vibrate out of the scala vestibuli and vibrate into the middle fluid-filled chamber of the ear. The middle part of the ear is called the cochlear duct. And the cochlear duct is where you can actually hear sound waves. There's still fluid inside the cochlear duct, but the fluid inside the cochlear duct has a different name. Its name is called endolymph fluid. Endolymph fluid. So the special fluid inside here is going to help the cells that actually do the process of hearing, help them to hear a sound. So let's review. Sound wave comes into the perilymph fluid up here in scala vestibuli. It finds the place along the cochlear duct where cells can hear that sound wave. It transfers from outside in scala vestibuli inside into the cochlear duct, where it moves the endolymph fluid in here and causes my cells to perceive the sound wave. We hear it here with cells we're going to remind ourselves of called hair cells. So hair cells get activated, they hear the sound wave. Now we're done hearing it. To make sure we don't keep hearing that sound wave, we send it out along the bottom here through something called scala tympani. So I've got a second fluid-filled tube down here. We talked about in lab how this one's kind of like a garbage disposal. This is my waste tube down here. So scala tympani is also filled with perilymph fluid. That perilymph fluid takes those leftover sound waves that I've already heard that I don't need to hear anymore and takes them over to a place called the round window. The round window is where sounds go to die. I spit them out here. There's nothing in the middle ear that's going to hear those sound waves, nothing to catch them. They just dissipate. They go away. So the process of hearing, we send sound waves through the air in the external middle ear. Then we send them through fluids, first perilymph fluid and scala vestibuli, then into endolymph fluid in the cochlear duct, and finally back out using that perilymph fluid that takes us to a place called the round window. This is where I get rid of sounds when I'm done hearing them. I've just said a bunch of things. I need you to help me out in the chat. What questions do we still have about this process before we move on? I got one thumbs up. Yeah, so it's easier to see on this picture, certainly. Yep. And I think I mentioned this when we looked at this picture for lab, but when you look at a picture of the internal ear, it doesn't look like this kind of long laid out thing here. It looks like a snail shell. If you imagine this tube curling and curling and curling on itself, that's what you see in the cochlea. So the cochlea looks like a snail shell because we take these three tubes and we curl them up on themselves over and over and over again. So when you're trying to translate this into what you normally see, that's how we would do that. There's a question in the chat about 4A, about what's happening right here in the picture. It's a great question. When we're talking about what's going on here at this part of the picture, you are surrounded by sound waves that you can't hear, either because they're too high pitched or because they're too low pitched. So if a sound wave comes into your ear and it's moving around, we go all the way down the cochlear duct, and there's nowhere on that cochlear duct where your ear can hear that sound wave. It will actually basically just recycle it. It'll go straight through your ears so you don't hear it at all. So for any sound waves that you can't hear, they'll go straight from scala vestibuli, where we're hoping that we can find a place to hear them. If we never find a place to hear them, we'll send them straight into scala tympani. So we'll just send them out the garbage disposal without ever actually hearing them. So when we talk about sound waves we can't perceive, we're going to bounce straight from scala vestibuli into scala tympani, and we don't hear them. When we talk about someone who's deaf, so when we talk about someone who is fully deaf, that's unlikely to be an issue so much with this, where we send our sound waves through. It's more likely that we've got an issue with their nerve, making it so that they can't hear, if they can't hear anything. Now, when you talk about people that can't hear high-pitched sounds or that couldn't hear low-pitched sounds, that's probably an example of a time where we're seeing more sound waves go through here. So if a particular section of their cochlear duct, the hair cells that live in here, don't work anymore, or the hair cells that live over here aren't working anymore, that would result in us just recycling those sound waves because the cells aren't working anymore. So partial deafness, we would probably see more sound waves going this way. With full deafness, again, that's probably an issue with the nervous tissue side of things. So sound waves are still doing this whole process, except when they go here into the cochlear duct, it doesn't matter. I don't hear them because something's wrong with my nerve or something's wrong with my neurons. And so this tyranny is less like you consciously ignoring something. It's more like how if you're ever sitting with your dog or your cat and they suddenly perk up and they like are whining or something, there's a sound in your environment that you can't hear that's bugging them, that's giving them trouble. It's more like that, where there's sound waves around you. It's not that you ignore them, it's just that you physically can't hear them. That's more what this is going on. So I guess kind of you could say your body ignoring them just because you don't have a way to hear them. So yeah, again, with animals is the best example. Their cochlear duct is a little more extensive than ours, so they can hear things that we can't hear. Okay, so let's take this picture and we'll rotate it to help us get to the other picture. Before I move on, let me read a quick question. So the question is, to clarify, perilymph moves the sound waves through the cochlear duct. Not quite correct. Perilymph moves the sound waves through scala vestibuli. So there's perilymph fluid here that moves the sound waves through scala vestibuli. When we get to the cochlear duct, to the right place where I could hear, the endolymph fluid translates those sound waves, and then we dump them back out into the perilymph fluid. Okay, yeah, you're perfect, Christina, but I'm glad we clarified. So Scala vestibuli, we move the sound waves, don't hear them, until we get to the place on the cochlear duct where I can hear them. Then the sound waves go into endolymph fluid. Once they're in the endolymph fluid and I hear them, I send them back out in perilymph fluid to transport them. So a good way to think about these two fluids in the body, perilymph fluid doesn't actually directly help you hear. The fluid that actually directly helps you hear is endolymph fluid. And the only place in the ear where you actually directly hear is this cochlear duct in the middle. Okay, so imagine with me that I took a knife and I made a cut right here in this tube, and then I rotated these 3 tubes around toward you. So I pulled them toward the front of the board. That's what we're looking at right here. So we saw those 3 tubes. Remember, the tube on top was scala vestibuli, then there was the cochlear duct, and then down below it was scala tympani. These are the 3 tubes that we saw in the previous picture. Now we're just looking at them, how they actually are in your body. They're stacked on top of each other. So sound waves first come into scala vestibuli. Remember, that's filled with the fluid that we called perilymph fluid. Perilymph fluid transports those sound waves. They're bouncing around inside here, bouncing, 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 until we find the place in the cochlear duct where we could actually hear them. At that point, the sound waves start to bounce around in the endolymph fluid in here. Now, here's the chemistry that we need to know about endolymph fluid. Endolymph fluid has a lot of potassium. It has a lot of K+. This is a note I want you to write down for yourself. It's something that I want you to remember. Endolymph fluid has a lot of potassium in it. Because when we start talking about the way that the hair cells that do hearing work, they're going to use this potassium to help them hear. So sound wave bouncing in perilymph, bouncing, bouncing, bouncing until we find the right place on the cochlear duct where the sound could be heard. Then we start bouncing around in this fluid called endolymph with a whole bunch of potassium in it. The sound waves that are bouncing around in the endolymph cause this bottom part of the cochlear duct, a part that we know from lab that was called the basilar membrane. Think about it like a basement, the basilar membrane. This membrane down here starts to bounce up and down with those sound waves that were in the fluid here. When this part starts to bounce up and down, here we're going to zoom in on this part right here. When the basilar membrane starts to bounce up and down, these hair cells that live on top of it, they start to bounce up and down as well. But remember from lab that on top of the hair cells, we have a thing called the tectorial membrane. The tectorial membrane. And this membrane does not move regardless of what's going on with the sound waves. So this membrane stays still. The basilar membrane, the basement that's holding these hair cells, bounces them up and down. These hair cells bump into the tectorial membrane. When these hair cells bump into the tectorial membrane, that's how you actually hear sound. So the bouncing of the basilar membrane that pushes these hair cells up into the tectorial membrane, that's going to activate your vestibulocochlear nerve. You'll hear a sound. Let's zoom back out. Here's those hair cells. They were bouncing up and down. They were hitting the tectorial membrane. But we've heard that sound now. I don't want to hear it any longer. This basilar membrane will actually transfer those sound waves down into scala tympani. Remember that scala tympani is where sounds go to die. We've got perilymph fluid that transfers those sounds back out to the middle ear where there's nothing there to hear them, and we get rid of them. So, process of hearing. We bounce around in scala vestibuli, which causes us to find the right place in the cochlear duct When I'm in the right place in the cochlear duct, the basilar membrane moves to make my hair cells get activated, and also to dump out those sound waves into the scala tympani, where I get rid of them. How do we feel about our 3 fluid-filled tubes in the cochlea? Which tube in the cochlear duct is the basilar membrane part of? So the basilar membrane kind of divides up the cochlear duct and scala tympani. So it's kind of the dividing line that's there in between them. So I don't know if we could say it's particularly part of one, because it literally is my dividing line between the two of them. Does that help, Nicole? Question on the worksheet. Okay, so if it's asking for a particular one, I would say, let's say it's part of the cochlear duct. That it's the bottom part of the cochlear duct that the hair cells sit on top of. Yeah, so it forms the bottom of it. Yep, so the cochlear duct, the basilar membrane does form the bottom where those hair cells rest on top of. Yeah. Okay. Here's a note that I want to make. I think I mentioned it in the guided lesson, but I want to emphasize it again on this picture before we zoom in even closer. When I ask you about the types of cells that do each of the special senses, the type of cells that do the sense of hearing are called hair cells. Hair cells. They get their name because if you look at them here on the top of these cells, they literally have little hairs. So little extensions, part where their membrane sticks out. These are the hair cells that are involved in hearing. Hair cells are going to pop up even more during our class this morning and our class tonight. So hair cells involved in almost all of the special senses. We have talked about the special sense that doesn't use hair cells. We've talked about how vision has a different kind of cells that helps it to work. What's the name of the kind of cells that help you to see? What are the 2 kinds of cells that help you to see? Yeah, Nicole's hesitant, but she's correct. The 2 types of cells that help you to see are the cells called rods and the cells called cones. Wink, wink, nudge, nudge. Remember that we need to know the difference between how rods and cones work. So when we talk about rods, they're really good at seeing in black and white. Rods can see when there's not as much light. But remember that rods are found more in what's called the peripheral areas of your retina, toward the outside of your eyes. So your peripheral vision, what you see not in the very center of what you're looking at, that's mostly the work of those rods that we have a ton of that live out there. Cones can see things really clearly. They see things in color, but they take a lot of light to be activated. Remember how we talked about that's why at night, when it's twilight, you can't really see in color. You just see in black and white. Because those cones say, can't see anything, don't know what's happening. So, friendly reminder, the cells of vision, called rods and cones, the cells of hearing, called hair cells. Hair cells. So let's look up close at one of these hair cells right here. This is what my picture shows me. We're looking up close at a hair cell. Now, remember when we were talking about our fluids, we said that endolymph fluid was what we find inside the cochlear duct, inside that tube where you actually do hearing. What did we say again that endolymph fluid had a lot of? What ion does endolymph have a lot of? Yeah, endolymph has lots of K+, lots of potassium. This is important because on these structures here, on these hairs, on the top of my hair cells, the parts that stick out, that give them their name hairs, we have mechanically gated potassium channels. Mechanically gated potassium channels. Hey, it's been a while since we talked about channels. We were all happy that we didn't have to talk about channels, right? But here they are back to haunt us. Mechanically gated potassium channels. First question about these channels. They are or are not always open. Mechanically gated potassium channels are or are not always open. Yeah, they are not always open. I know that because they're gated. Gated means I'm not always open. Yeah, and Tierney is totally right. When we talk about mechanically gated channels, we're talking about how we get them open with this mechanically word here. Mechanically gated channels were the ones that we had to push open. Mechanically gated channels. Hey, so check this out. I'm a hair cell. I have channels that you have to push open. When I push them open, potassium moves, and in endolymph fluid, there's a bunch of potassium outside. So that potassium is going to come inside of me. To allow the potassium to enter my cell with these mechanically gated channels, I've got to push on them. What was the name of that thing that's up here above the hair cells that we said doesn't move when sound waves are in the cochlear duct? Does anyone remember its name? Yeah, it's that thing on the top called the tectorial membrane. The tectorial membrane. I'm going to kind of pretend to draw it up here. The tectorial membrane was that thing that was on top of, it kind of looked like it had little fibers in it. It was that thing resting on top of the hair cells that didn't move. The function of the tectorial membrane is to push open mechanically gated potassium channels. Because see that basilar membrane that's underneath the hair cells, when that membrane starts bouncing up and down, these hair cells start bouncing up and down, but the tectorial membrane doesn't move. So these hairs bump into it, and when they bump into it, we push open mechanically gated potassium channels. So the tectorial membrane is what's going to give me that push to open my mechanically gated potassium channels. Potassium rushes inside because there's a whole bunch of potassium in the fluid out here. Now notice when potassium rushes inside, that depolarizes my cell. Let's do a review here, especially since we have that final exam coming up, right? When I use this word depolarization, what does depolarization mean? What's going on? Yeah, so depolarization means I'm making my membrane more positive. Membrane more positive. When potassium rushes inside a hair cell, the membrane charge gets more positive. That's because potassium has a positive charge. What's the name of an ion that has a positive charge? Positively charged ions. Yeah, those are called cations. Cations. Potassium is a cation. has a positive charge. Because there's so much of it outside, it wants to come inside to this cell. It brings its positive charge with it into the cell. And check this out. When inside this cell gets positive charges, voltage-gated calcium channels open up. Voltage-gated calcium channels. Let's do my favorite questions again. Voltage-gated calcium channels are or are not always open. Voltage-gated calcium channels. Yeah, again, we've got that word gated. Again, we know this channel is not always open. So when I make my membrane more positive, when I change the voltage, my voltage-gated calcium channels open up. In comes calcium. And just like with my neurons, my regular neurons that we talked about back in unit number 3, when I bring in calcium into a hair cell, that causes the hair cell to spit out neurotransmitters. Remember, calcium and neurotransmitters, they don't mix. So if calcium comes inside that hair cell is going to spit out neurotransmitters. Those neurotransmitters go down and talk to the neighboring neuron and tell it, we're hearing a sound. So the process of hearing a sound is very similar to the process that we used in normal neurons to talk to each other. The biggest difference is right here. The process of hearing a sound starts with a mechanically gated potassium channel. When I press on these channels and let in potassium, my membrane gets more positive, which causes me to bring in calcium. And when I bring in calcium, I spit out neurotransmitters to tell the brain that I'm hearing something. The question in the chat is, do we need to know the voltage that these voltage-gated calcium channels open at? No, we do not, because these voltage-gated calcium channels, as we're going to see in a moment here, they can open a lot or they can open just a little, as long as you know in general that these voltage-gated calcium channels, they're only going to open if the membrane has started to become more positive. As long as you know that they're opened by a more positive charge than resting membrane potential, you're good. So just know that it takes a positive charge to open them. We don't need to know a number on what opens these ones. Okay, so let's look at more in depth here. Let's look at how these mechanically gated channels lead us to do the process of hearing. I want to start with the basilar membrane at rest. The basilar membrane at rest. What this picture is showing me is when you're not hearing a particular sound, when you're just listening to the environment around you, what's going on with your hair cells. So in normal situations, your hair cells are sitting on top of the basilar membrane. The basilar membrane is flat. We are standing upright. In normal situations, the tectorial membrane just barely touches the tallest hair on our hair cells. That tallest hair is called a stereocilia. So the tallest hair touches the tectorial membrane just a little bit. Because it touches that membrane just a little bit, notice that we have maybe one or a couple of our mechanically gated potassium channels pushed open, because the tectorial membrane is just barely touching my tallest hair. So when you're not hearing a sound, we're touching that membrane just a little bit. We let in a few potassium ions. When we let in a few potassium ions, they make our membrane charge a little bit higher than normal. If it's a little bit higher than normal, we let in a little bit of calcium down here. and we spit out a little bit of neurotransmitters. Hair cells can send out different numbers of messages. And an important idea about the way that you hear things. Hair cells send out different numbers of messages. When we're not hearing a sound, so we're not particularly hearing something at one pitch, we will send out little messages, just a few neurotransmitters back to the brain, so back through the neuron, letting the brain know, I'm listening, but I don't hear anything. I'm listening, but I don't hear anything. We'll keep sending this little steady stream of signals to the brain saying, we're listening, but we don't hear anything. Hey, check this out. We talked about this. What is the part of your brain that takes these little messages and tells the brain, we're listening for something. We don't hear it yet, but we want to keep listening? What's the part of your brain that primes it or keeps it ready to receive more information? Yeah, so one of the places, there's a couple of places that we mentioned in the chat that we'd send this information. One place we'd send it would be to the reticular activating system, the part of your brain that sends out pings, remember, to keep the auditory area awake. So we send out a few messages, hey, listening area, we're still listening here, we're still trying to maybe hear something. But the other place that we'd send it to, the fact that these aren't messages that say we're hearing a sound, they probably consciously would get filtered out by the thalamus, like we've mentioned. So the thalamus might hear these little messages or see these little messages. Oh, we're not actually hearing anything. You don't consciously process it. But it also keeps that reticular activating system ready to hear more signals. The basilar membrane at rest. When we aren't hearing a particular sound. When we're not hearing anything in particular, your basilar membrane is not moving. It's completely flat. The tallest hair on a hair cell is touching the tectorial membrane, which means that we've got a few of those mechanically gated potassium channels that are open, which means we're letting in a little bit of calcium in here, which means we're sending out a little bit of neurotransmitters. Let's switch to our middle pane here. When our hairs are bent toward the tallest hair or the tallest stereocilium, this is what happens when you're in the process of hearing a sound. So when we are actively hearing that noise of a car driving by, of my daughter screaming, my favorite sound, when you're actively hearing a sound, at that point, the basilar membrane is bouncing or it's tilting toward the tallest part of the hair cell. Now, let me mention something. I'm not going to ask you on the exam which direction the cell is bending. I want you to know that the basilar membrane, in this case, we're bouncing up and down. So we're bouncing up because we're hearing a sound. When we bounce up, that causes the hairs, the things on the top of here, the whole cell gets pushed even closer to that tectorial membrane. When I push all of those hairs against the tectorial membrane, I open up all of those mechanically gated calcium channels. So I open up all of those mechanically gated calcium channels, in comes my potassium. As my potassium rushes in's going to come some calcium as well from my voltage gated calcium channels. Calcium comes inside and my hair cell spits out tons and tons of neurotransmitters. The way you perceive a sound is because my hair cell sends out all kinds of neurotransmitters that my neuron receives and perceives all of them. So notice that I've got a lot of messages that came from all of these neurotransmitters. As those neurotransmitters cause this neuron to receive the messages, ultimately your brain will interpret that as I heard something. So lots of neurotransmitters come out. I am hearing something as we speak. Just a few neurotransmitters. I haven't heard anything recently. I'm waiting to hear something. Because see, check out over here. When you just finish hearing a sound, so after you've perceived and spit out all of these neurotransmitters, because the basilar membrane was bouncing, the basilar membrane then sends those sound waves down into scala tympani, down below it. The basilar membrane will actually bend the other direction when you're done hearing a sound wave. So once you've perceived it, the basilar membrane bends the opposite direction. And when the basilar membrane bends the opposite direction, it pushes closed the mechanically gated potassium channels. Now we're in the wrong shape completely, where I've completely slammed those gates shut. Nobody's being pushed open because of the direction of the basilar membrane, because it's sending those sound waves away. So when I'm just finished hearing a sound, we're recycling sound waves. All of my mechanically gated potassium channels get pushed shut. If all of them are pushed shut, no potassium is coming inside. If no potassium is coming inside, then no calcium's coming inside. And if no calcium is coming inside, no messages are being released. If I release no messages, my nerve never gets activated. There's no sounds at all. This is the way your brain knows you just stopped hearing something. So if we think about the process of hearing, we start with a whole bunch of neurotransmitters spitting out, sending a ton of messages, to no neurotransmitters being spit out when we just finish hearing a sound, to going back to just a few little pings being sent out while we wait to hear another sound. So when you hear something, lots of neurotransmitters being spit out. When you just finished hearing something, we send no neurotransmitters to help the brain know that we stopped hearing that sound. And as we return to normal, when we're waiting to hear the next sound, we spit out just a few neurotransmitters. On the homework assignment, you have some practice questions that ask you about how many neurotransmitters are being spit out. So we spit out just a few when we're waiting to hear something. We spit out a ton when we've just heard something. We spit out none when we just finished hearing something. What questions do we have about neurotransmitter release and the process of hearing? Got one thumbs up. Okay. Well, as you keep working on it and you keep working on the homework assignment, if you have more questions about it, please feel free to bring them to our review session, or feel free to reach out via email. The good news is, if you have a general understanding of the way that hearing worked, we use a similar process when we're talking about equilibrium, when we're talking about balance. So when we talk about equilibrium, or the normal person word for it is balance, we are still going to be using hair cells. We're still going to be using mechanically gated channels. And while we're at it, we're still going to use endolymph fluid. So many things are the same when we talk about the process of balance. When we do balance, we're talking about parts of the ear called the saccule and the utricle. Hopefully remember those from lab. And we're talking about these parts up here called the semicircular canals. When we do the process of equilibrium, there are two kinds of equilibrium or two kinds of balance that we do. The first kind is called static equilibrium. Static equilibrium. And the technical way to describe what's going on with static equilibrium is it's your position relative to gravity. Relative to gravity. In other words, it helps you to know kind of if your head's going forward or backwards. Forward or backwards is a good way to think about it. For my friends who've had a chance to work on this part of the lesson, do you remember which part of or which parts of the ear help us with static equilibrium? Which parts do static equilibrium? So forward and backwards or relative to gravity. Yeah, so the two parts that do static equilibrium are the utricle and the saccule. The utricle and the saccule. So these two fluid-filled sacs, here's what we just talked about with hearing, our cochlea over here. Here's where hearing happens. Here's where static equilibrium happens, in the saccule and the utricle of the ear. So keeping you from falling forwards, from falling backwards, or a good example of when you feel static equilibrium is when you're in an elevator and you start to feel the elevator go up or down. That's you doing static equilibrium, making sure you don't fall forwards or backwards when gravity changes. So static equilibrium done right here by the saccule and utricle. We also have what's called dynamic equilibrium. Dynamic equilibrium, or the easier word for it is rotational. Rotational equilibrium. Rotational equilibrium is done by the semicircular canals. Semicircular canals, that's these three things that we have here in the ear. When we're talking about precisely how your body is rotating, we need to be able to detect rotation in all kinds of different directions. We need to be able to detect leaning in all kinds of different directions. So we have three semicircular canals to help us know exactly how we're rotating. So big idea here, equilibrium means balance. When we talk about rotating balance, that's these three things here on the outside, the semicircular canals. When we talk about forward and backwards, relative to gravity, that's the saccule and the utricle that are here, closer to the cochlea. So let's start with static equilibrium. Static equilibrium is the one that we said was done by the saccule and the utricle. When you're thinking about the types of equilibrium, a really good study pro tip for you would be to make lists of the structures that are involved in them. So the first structure that is involved in the process of static equilibrium is something called the macula. Now, this is a word we already mentioned today in a different context. What was the other thing with that word macula in it that we mentioned today? Let's make a note for ourselves so we don't get confused. Yeah, we talked about the macula lutea. What was the deal with the macula lutea? What was going on there? Or what is the macula lutea? Yeah, this is where we have the clearest vision. Clearest vision. Oops. Try that again. Clearest vision. This is your place in the eyeball where we have the most cones. We have the most cones at a place called the macula lutea. So first time we talk about macula, when we're talking about vision, the macula lutea. Second time we're talking about the macula today, we just call it the macula. The macula of the saccule and the utricle, this is the membrane that detects Static equilibrium. Static equilibrium. We're getting into some really dicey spelling words here, and I totally butchered that. I apologize. Or pretend that actually says equilibrium. So the macula of the saccule and the utricle, that is the structure we're going to use to detect if your head's bending forward or bending backwards. So here's kind of generally where I find it in the saccule and the utricle. If I zoom in close on it, I can see that there are several parts to the macula that makes up this detecting part in the saccule and the utricle. Here's an underline highlight star word because Dr. Ellis just likes it. It brings me joy in these hard times. I really like otoliths. For whatever reason, otoliths always make me happy. So otoliths are like little crystals, little calcium carbonate crystals. We find them inside the saccule and the utricle. They sit on top of this jelly membrane here, or a jello membrane, if you will, called the otolithic membrane, or the otolith membrane. These little otoliths on top of it, sometimes they kind of look like little snowmen, or my daughter is really into Frozen, so they kind of look a lot like, she calls him Olaf. For any not quite as Frozen obsessed as we are over here, his name is actually Olaf, but my daughter calls him Olaf, and I will never correct that. So we got little Olafs here, or little Olaf the snowman. These things are called otoliths. When we're doing the process of determining whether your head goes forward or backwards, it's going to be the movement of these little otoliths that allow us to detect that. So anytime you hear otoliths, you should be thinking static equilibrium. You should be thinking head going forward or backwards. Anytime you hear macula by itself, not macula lutea, just macula, we should be thinking static equilibrium, head going forward or backward. And anytime you hear utricle or saccule, you should also be thinking static equilibrium. So these are the big structures of static equilibrium, those otoliths that I find inside this membrane called the macula lutea. Notice that the specific type of cells that are going to detect whether your head's going forward or backwards, they're all called hair cells. So hair cells, just like they were involved in hearing, they're involved in equilibrium as well. Let's talk about how you know if your head's going forward or backwards. Static equilibrium. Normal situation, head upright. When my head is upright, those otoliths, those little Olaf snowmen here, those otoliths are resting on top of, straight on top of the hair cells. The hair cells are covered in those mechanically gated channels, like we talked about with hearing. When otoliths sit straight on top of them, some of those channels are open. So my neurons, my hair cells in the macula send out kind of a normal number of messages when a steady stream, it says, of messages when my head is upright, when I'm not falling asleep in class, right? That's what I like to think about static equilibrium, the two ways you fall asleep in class. When I'm awake and alert, when my head is upright, my hair cells send out a steady stream of messages saying we're not bending forward, we're not bending backwards, because all the otoliths are just sitting straight on top. When I start to nod off with my head going forward in class, those otoliths, because gravity's pulling on them, the otoliths start to bend forward. When the otoliths bend forward, they push open those mechanically gated channels, like we talked about with hearing. The otoliths scoot down, push open those channels, in comes potassium, out goes neurotransmitters. When your head starts to go forward, when you're nodding off in class toward the front, you should send a bunch of messages back to your brain saying, we're leaning forward, oh no. But the funny one, and we've all probably seen this in class at some point in our life, is when someone nods off toward the back, right? When you're completely exhausted, this is the way it goes. This was snoring. Our otoliths go backwards, closing those mechanically gated channels on top of the hair cells. When those channels get pushed closed because the otoliths are going backwards, that makes us send out fewer messages to the brain. Our brain interprets fewer messages from the macula to mean that the head's going backwards. So the way that you figure out where your head's at, if it's straight up, if it's bending forward, or if it's bending backwards, the way you figure that out is by the number of messages that your hair cells are sending. If I'm sending a normal, steady number of messages, like I see here, that lets me know my head is upright. If I'm sending a ton of messages, that tells me that my head is bending forward. If I'm sending just a few messages, that tells me that my head is bending backwards. So we use the rate of messages that we're receiving to help us know if we're going forward or backwards. Just like, remember, with sound, we use the rate of messages we're receiving too. Remember, with sound, if we had just a few steady stream of messages, that was, I'm listening, but I don't hear anything. We would have a ton of messages when we were hearing things, and we actually heard nothing or no messages when we just stopped hearing something. So it's all about those rate of messages in the process of hearing and in the process here of static equilibrium. Let's talk dynamic equilibrium. That's rotational equilibrium. When we're talking about rotational equilibrium, the words that should always tip that off in our mind, the first word is something called the crista. The crista is the nervous tissue structure that I use to help me detect rotational movement. So kind of like how we had the macula when we were doing static equilibrium, we have the crista in dynamic equilibrium. Dynamic equilibrium is detected in these loop things that we call the semicircular canals. So the semicircular canals, the tubes, each have a crista, that's this membrane I see down here, that helps me to detect which way I'm rotating. On top of the crista, where I've got my neurons, where I've got my hair cells, on top of the crista, we have this little thing here called the cupula. It kind of looks like a tongue of fire to me. So we've got a little bit of fire up here, like on top of a candle. This thing called the cupula. When we think about you detecting which way you're rotating around, the way we're going to detect that is how bent this cupula structure is. So let's just go to our picture here to look at it. Before we start spinning, when we're just standing upright, our cupula is not moving. So in one of my semicircular canals, that cupula is just standing upright. It's waiting for us to move. When your body starts rotating, or in this extreme example with an ice skater here, when we're spinning around rapidly in circles, the fluid, the endolymph fluid that we have inside the semicircular canals, starts to move in the direction that we're rotating here. So the fluid starts to move. Well, to be more accurate, your body moves before the fluid does. But the big thing for us to know here is the cupula gets bent. When we start moving, the cupula is bent because the fluid slowly starts to move. As the fluid slowly starts to move with the body that's moving faster, that bends the cupula. Cupula gets bent. We're sending a bunch of signals to the brain, telling the brain that we're detecting rotational movement. When we stop spinning in a circle, that fluid is still kind of moving, but our body isn't moving anymore. We actually detect it as bending the cupula the opposite direction. So then we're going to send the opposite directions, or we're going to stop sending messages to the brain. That's how our brain would interpret that we are no longer spinning. So that period when you spin in circles for a whole bunch of time and then you stop and you're like so dizzy and you can hardly keep your balance, it's because that fluid inside the semicircular canals, it's not that the fluid stops moving. The fluid keeps moving a little bit longer, and your body, your brain in particular, has to recognize we've stopped spinning, even though my ears feel like we're still spinning. We're spinning the opposite direction. So in the process of rotational equilibrium, when we're not rotating, when we're standing still in the ice skates, the cupula is straight up. We start rotating, the cupula bends one direction because we are rotating, because our fluid is moving in the tube. We stop moving, our cupula gets bent the opposite direction because the movement has stopped. What questions do we have about dynamic equilibrium? Anyone feeling dizzy? Is your mind feeling dizzy with all these processes this morning? So Christina asked if there's a specific reason that it's endolymph and not perilymph. So the structures or the fluid that's around my hair cells, the fluid that helps my hair cells to do their job is endolymph fluid, because hair cells need to be depolarized with potassium. So endolymph fluid has a lot of potassium in it. Perilymph fluid actually does not. So I need to have endolymph fluid inside these semicircular canals to make sure that I have the potassium that I use to depolarize the hair cells. So it's because of the chemistry that makes the fluids different. Perilymph fluid doesn't have enough potassium in it to get your hair cells excited. That's why I have to have it. Yeah, and to Gloria's question, that's absolutely right. When you're not moving, the fluid is standing still. When you're not rotating, but the caveat to that being, when you first stop rotating, your fluid rotates more slowly than your whole body does. So if you notice, I know I saw it down here. Where we go? Right, yeah, right here. Here's your chemistry physics word for the day: inertia. So when you first start rotating, your fluid inside these semicircular canals is not moving. It's inert, it's standing still. You start rotating your body, your fluid doesn't start rotating at the exact same time. So that's why we see the fluid, it kind of looks like it's flowing this direction. Technically what's happening is the cupula is moving this direction because we're rotating, and the fluid is standing still. When we first start rotating, the fluid doesn't start rotating with us right away. That's why we detect that we've started rotating. Eventually, we'll get to an equilibrium point where our fluid is moving as fast as our ear is moving. And that's kind of when you hit that sweet spot, like when you're on a merry-go-round and like you don't feel the movement as much anymore. You don't feel like you're rotating. But when you stop rotating your body, that fluid is still moving around in circles like you were. So see, you were moving in circles this way. The fluid keeps going for a while. Again, because of inertia. The fluid is just going to go and go until it runs out of that energy to keep moving. The way your brain interprets that, though, is I stopped moving, that fluid is moving that direction. That inhibits me. I stop sending out those messages to the brain. So I know that my body has stopped moving, but I'm gonna still be wobbly for a little bit. As long as that fluid is still moving in my ears, as long as it's pressing on those hair cells and slamming them shut, I'm going to feel a little bit off kilter, a little bit off balance because that fluid is still moving. Once it stops moving, then I'll feel normal again. When my fluid's still, I feel normal, the cupulla's straight up. Otherwise, I'll feel like I'm rotating, or I'll feel like I just stopped rotating, kind of like with hearing the sounds. Okay, so let me give us an option here. Options are either I briefly talk about smell and taste, because those are really short. I could talk about them now and we could use tonight to review, or we can just call it a day now and I can save those things for later. What would we prefer? Got a couple of votes to just plug through them, taste and smell. They're really easy, especially compared to all of this all of this dynamic and static equilibrium stuff. They're going to be super easy. So perfect. We'll go ahead and do it really fast. And then tonight, whatever we want to chat about, we can, or we can not. We can decide. When we talk about the process of smell, we're talking about olfaction. Olfaction, the process of smell, happens inside your nasal cavity. It's actually about only 1 square inch of your entire nose that helps you to smell. It's not a lot of space that helps you to smell. When we talk about smelling, the type of cells that help us to do it are called olfactory hair cells. Olfactory hair cells. Olfactory hair cells are these little yellow cells that we see that extend down from the olfactory bulb, or the olfactory nerve, cranial nerve number 1. We send down extensions down into an area called the olfactory epithelium, so that little square inch section in your nose. At the very end of these hair cells, we have our olfactory hairs, the extensions that actually do the process of smell. We call the process of smell a chemical sense, a chemical sense. This is not a trick question. If this is a chemical sense, what are we actually detecting in the process of smelling something? What are we actually smelling? Yeah. We're chiming in slowly. What we actually smell in the process of olfaction are chemicals. We smell chemicals. And those chemicals have a special name. The chemicals that you smell are called odorants. For you to be able to detect an odorant, we must dissolve it. So the chemicals that you smell have to be dissolved for you to be able to smell them. What we dissolve them in is mucus. So the purpose of the mucus in your nose is to help you dissolve the chemicals or dissolve the odorants that you're smelling. Hey, let's make a guess here, an informed guess. We have a particular type of gated channels that live on my olfactory hairs down here. Which kind of gated channels do you think we might use to help us to detect odorants? What kind of gated channels? Yeah, exactly. On my olfactory hair cells, on their hairs, we have chemically gated ion channels. Chemically gated ion channels. These chemically gated channels, when the chemical we're smelling attaches to them, it activates them. And we detect that activation with these olfactory hair cells that take the sense of smell back up into the olfactory nerve and send it back to the brain. In the guided lesson, it asks you to consider why we sometimes can or cannot smell when we get a cold. It has to do with the amount of mucus that your body makes. See, there's this fine line for the amount of mucus that we need. We need enough mucus to cover the olfactory hairs so that we can dissolve our chemicals, we can dissolve those odorants so they can be detected by our chemically gated ion channels. We need enough mucus to do that. But if we have too much mucus, I might dissolve those chemicals, but if I've got mucus that goes all the way down here, say I've got a really bad cold and I've got a ton of mucus down here, I might dissolve something in the mucus that's down here, but there's no olfactory hairs down here. And if there's no olfactory hairs, I can't detect those chemicals. So we need the right amount of mucus, not too much, where I'd never get those things up to the hair cells, not too little, where I couldn't dissolve my chemicals. I need the just right amount of mucus to help me so that I can detect those chemicals in the process of smell. So the chemical sense of smell, the chemicals we're detecting are called odorants. In the chemical sense of taste, the chemicals that I'm detecting, so we'll write here it's a chemical sense, and the chemical sense of taste, the chemicals I'm detecting are called tastants. Again, because it is a chemical sense, I have to dissolve my chemicals to be able to detect them. What do I use in the chemical sense of taste, also known as gustation? What do I use in the chemical sense of gustation to dissolve my chemicals? What do you use in your body to dissolve tastants? Yeah, that's the job of saliva. The reason we make saliva is to help us to dissolve the chemicals that we're tasting. We dissolve them on the surface of the tongue, like I see right here. Then those dissolved chemicals in saliva go down into little holes on the surface of your tongue, where they go down and meet gustatory hair cells. Remember, I told you at the beginning of class today, hair cells are going to be all over the place. The process of gustation, or the process of tasting, also involves hair cells. So at the very end of these hair cells, we have gustatory hairs. Gustatory hairs are the little extensions that are activated by the chemicals, the tastants that are dissolved in saliva. Here's my question again, not a trick question. Which kind of gated channels are going to be on gustatory hairs? What kind of gated channels help me to do taste? Yeah, again, it's going to be chemically gated channels. Chemically gated channels. So chemically gated channels help us to do the process of taste. Chemically gated channels help us to do the process of smell. The process of taste is about 80% smell. So if you can't smell, you can't taste. But here's Dr. Ollis's hack for you. Next time you get a cold and you can't taste, if you blow your nose, you might be able to taste for just a moment. Let's go back here to smell. If you blow your nose, you might be able to taste for just a moment. Why might that be the case? Yeah, so Christina is mentioning when we blow our nose, what we're doing is we're getting rid of mucus. If you have a cold, you make a ton of mucus. So much mucus that, like we were talking about, our chemicals get dissolved down here and they never make it up to our olfactory hairs. If they never make it up to our olfactory hairs, then we're never going to detect them. If I get rid of some of this mucus, that would allow those odorants to get up here to the nose. And remember, odorants are what help us to smell, but smell is the majority of what we taste. If I can help my smelling process work better, I will be able to taste better as well. So there's how you hack your cold to be able to taste for just a brief moment of bliss, right? It's so sad when you get a cold and then you can't taste anything. So there's your bliss moment there. Yeah, too much mucus makes it difficult to smell. Exactly. Absolutely. That is lesson 13. Look at us. We wrapped it up earlier than I expected to. So congratulations on getting through all of the material in Anatomy and Physiology 1. If I was there with you, I would give you a high five. So virtual high five. Let's see if we got a little high five emoji going on here. That's not working. Here, let's try this. Here we go. I'm gonna give you a fist bump. I'm giving you a fist bump. My 4-year-old can do fist bumps. So fist bump, high five to you guys. I'm really proud of you. We have gone through all of our material. So I'll open the floor for questions here, and then I'll plan for us to maybe do an application activity tonight, just to kind of do some group work, and I'll be open to answering any questions that we have. So let me look at Jacqueline's question here. Yeah, so Jacqueline is asking, because there's a question on the outline, asking you about having too much or too little saliva. Christina's absolutely right. The process of taste works pretty much just like the process of smell. So I need the right amount of saliva to dissolve my tastants. I dissolve them, then they can go down in my little taste pores to get to my hair cells so that I'm able to detect them. If I have too much saliva, just like if I had too much mucus, those little tastants would be out here on the surface of the tongue and they'd never go down into my taste pore. If I have too little saliva, those tastants would stay on the outside of my tongue and I wouldn't detect them either. So I need to have the right amount of saliva to dissolve my chemicals, but not to dilute them, basically, just like with the process of smell. Yeah, so Nicole asked about our timing tonight. Yeah, 5 o'clock tonight. We'll bring some questions for us to work through. We can talk about some of those critical thinking things, and review any other topics from unit number 4 that we want to chat about. Any other last minute questions? Let me give you a penguin, because it's been a little while since we had a penguin. Although I think some of us, a couple of us saw my little glasses penguin that I made after most of us left last time. Give you a penguin here. He's excited. I'm going to give him a party hat, because we're anatomy rock stars. Look at us. Perfect. Yeah, you're welcome, Tierney. I'm happy to help you all out. All right, well, I'm going to stop the recording. I will stick around for a couple minutes if anyone has questions. Otherwise, I hope to see several of you back tonight. Otherwise, we will do our organized big Unit 4 review on Wednesday. So if you aren't here tonight, I will see you on Wednesday.